Polyphenol, the diverse and powerful class of plant-derived compounds, have captured the attention of researchers for their remarkable ability to support cellular health, reduce inflammation, and promote longevity. Among their most intriguing and biologically valuable properties is their capacity to modulate autophagy, a fundamental cellular process responsible for cleaning up damaged structures, maintaining protein quality control, and preserving overall cellular homeostasis. As scientific understanding of the mechanisms of aging and chronic disease deepens, polyphenol are increasingly being recognized not just as antioxidants or anti-inflammatory agents, but as direct influencers of gene expression and signaling pathways tied to cellular self-renewal. One of the key pathways through which polyphenol exert their beneficial effects is the activation of sirtuin-1, SIRT1, an enzyme that plays a central role in longevity and metabolic regulation. SIRT1 is a NAD-dependent deacetylase that regulates numerous downstream targets involved in stress resistance, DNA repair, mitochondrial biogenesis, and notably autophagy. When polyphenols such as resveratrol, quercetin, and EGSG are introduced into the cellular environment, they enhance SIRT1 activity, leading to the deacetylation of several autophagy-related proteins. This biochemical modification facilitates the formation and elongation of autophagosomes, the vesicles that engulf cellular debris for degradation. By enhancing this pathway, polyphenol essentially help reboot the cell's internal cleaning system, promoting more efficient repair and turnover. In parallel, many polyphenol exhibit anti-inflammatory effects that further support autophagy. Chronic low-grade inflammation, often referred to as inflammaging, is known to suppress autophagic activity and contribute to cellular dysfunction over time. Polyphenol counteract this by inhibiting NF-B signaling, a key pathway involved in the production of pro-inflammatory cytokines. By reducing the inflammatory burden, these compounds create a cellular environment more conducive to effective autophagic recycling. In this way, polyphenol don't just trigger autophagy, they remove one of the biggest barriers to its optimal function. Among the most widely studied polyphenol is resveratrol, a stilbene found in red grapes, berries, and peanuts. Resveratrol has been shown to mimic some of the effects of caloric restriction and fasting, both of which are strong inducers of autophagy. It enhances mitochondrial function, improves insulin sensitivity, and promotes survival pathways under stress conditions. In multiple animal models, resveratrol supplementation leads to increased autophagy in the brain, liver, and heart, offering protection against neurodegenerative diseases, fatty liver, and cardiovascular disorders. Quercetin, a flavanol abundant in onions, apples, and capers, also demonstrates strong pro-autophagic effects. It not only activates SIRT1 but also modulates AMPK, another master regulator of energy metabolism and autophagy. Quercetin has been shown to protect cells against oxidative damage and enhance autophagic flux in models of aging and metabolic disease. It supports the clearance of dysfunctional mitochondria and the reduction of cellular senescence, making it a key candidate in the effort to develop dietary strategies that slow aging at the cellular level. Another exceptional polyphenol is epigallocatechin gallate, EGSG, the major catechin found in green tea. EGCG has demonstrated the ability to upregulate autophagy-related genes while also reducing oxidative stress and lipid peroxidation. It is especially noted for its neuroprotective effects as it promotes autophagic degradation of misfolded proteins in the brain, an essential function in preventing conditions like Alzheimer's and Parkinson's disease. Additionally, EGSG has been shown to inhibit mTOR signaling, a pathway that, when overactive, suppresses autophagy and accelerates aging. By delicately balancing energy sensors and stress responses, EGSG helps the body maintain an efficient cycle of renewal. What is particularly exciting about polyphenol is their availability and versatility. Unlike pharmaceutical interventions, these compounds are found in common foods and beverages that can be easily incorporated into the diet. Drinking green tea, consuming fresh berries and red grapes, cooking with onions and herbs, or enjoying a moderate amount of dark chocolate all contribute to a polyphenol-rich dietary pattern. When consumed consistently and as part of a whole food, plant-forward approach, these compounds can provide continuous, gentle stimulation of autophagy pathways without harsh side effects or metabolic stress. Moreover, Polyphenol often work synergistically with each other and with other autophagy-inducing strategies such as intermittent fasting, caloric restriction, and ketogenic diets. For example, combining resveratrol supplementation with a low-calorie or fasting protocol may amplify the activation of SIRT1 and deepen the autophagic response. Likewise, EGSG taken during a fasted state may improve metabolic flexibility while enhancing the clearance of damaged cellular components. In essence, polyphenol offer a uniquely elegant solution for promoting long-term cellular health and resilience. They engage the body's own systems of repair and maintenance, particularly through the upregulation of autophagy, while also providing protection against oxidative stress and chronic inflammation. As research continues to unravel the molecular underpinnings of health span and aging, polyphenols stand out as powerful, natural tools for enhancing the body's intrinsic capacity for renewal and protection. Their accessibility through everyday foods only adds to their appeal, making them a practical and potent ally in the pursuit of longevity and vitality.